Hello, class. This is a discussion on PhysioEx exercise 1. In exercise 1, we did activities 1, 2, 3, and also activity 5. So 1, 2, 3, and 5. The first activity, activity 1, we were simulating dialysis. This was supposed to show us simple diffusion. If you look at the video playing in your top right corner, you're seeing solutes moving across a cell membrane through channels. These channels are not gated. They allow permeability for specific solutes. We were trying to simulate the same type of thing that we see in a living biological membrane. In the bottom right picture, you see the apparatus that we used for this activity. We had 2 chambers, and in between the chambers, we had a dialysis membrane. That dialysis membrane was supposed to represent a biological membrane. This dialysis membrane had a molecular weight cutoff associated with it. What does that mean? Molecular weight cutoff refers to the pore size, the holes in that dialysis membrane. If a solute was smaller than the molecular weight cutoff, then the solute was able to move across that dialysis membrane. Let's review what you did. So we actually did an experiment very similar to this in our lab. We used dialysis membrane, we filled it with solutes, and that dialysis membrane that we played with in lab had holes in it. In this PhysioEx, we were playing with 4 different molecular weight cutoffs: 20, 50, 100, and 200. We also played with 4 different solutes: sodium chloride, urea, glucose, and albumin. Now, sodium chloride has a molecular weight of 58.45. Urea has a molecular weight of about 60. Glucose has a molecular weight of 180. And albumin is in the thousands of kilodalton size. So the question was, when would these solutes be allowed to move? When could they diffuse through those holes? So in my picture over here in the magenta, I'm showing the holes in the dialysis membrane. And in blue dots, I'm showing you small solutes. And with the blue square, a very large solute. We started to collect our data. These data are in units, diffusion rate units of millimolar per second. So first we used sodium chloride, and we used a molecular weight cutoff of 20. Well, sodium chloride has a molecular weight of 58.45. So clearly, sodium chloride is much too large to get through that molecular weight cutoff. Urea, having a molecular weight of around 60, also was not able to get through a molecular weight cutoff of 20. So then we moved on to a molecular weight cutoff of 50. And our first experiment with a molecular weight cutoff of 50, we used a 9 millimolar sodium chloride solution, and it actually diffused. Why did that happen? If sodium chloride has a molecular weight of 58.45, it should not have been able to get through a molecular weight cutoff membrane of 50. Or can it? Are you not remembering that sodium chloride dissociates? You already have had a chemistry and metric exam, and you should remember that sodium chloride has a dissociation factor of 2. Chloride has an atomic weight of around 35, and sodium has an atomic weight of around 23. So 23 and 35 individually clearly are below the molecular weight cutoff of 50. We repeated the sodium chloride experiment a second time, but this time we used 18 millimolar. And look what happened. Our rate of diffusion pretty much doubled. Why is this? This refers, or this is a concept called Fick's law for the rate of diffusion. The rate of diffusion, according to Fick's law, and we're just going to address a small part of Fick's law. When we get to Unit 4, we will definitely learn all the parts of Fick's law. But for right now, you should know that Fick's law for the rate of diffusion is directly related to the concentration gradient. If the concentration gradient goes up, so will the rate of diffusion. Fick's law is also directly related to surface area. Well, surface area did not change in this experiment. That was held constant. We will address surface area in lecture unit 4. Fick's law also says it's inversely related to distance. The greater the distance, the reduced rate of diffusion. We will also come back to that concept in lecture unit 4 when we learn how the respiratory membrane can actually have changes in its thickness, and this reduces the rate of oxygen diffusion in certain patients. So surface area was kept constant, distance was kept constant, and we can see that the 9 millimolar versus 18 millimolar, when the concentration went up, so did the rate of diffusion. Then we went to a molecular weight cutoff of 100, and we were using 9 millimolar sodium chloride again. We got the same rate of diffusion as we did for 9 millimolar using the 50 molecular weight cutoff. Well, it moves the same through a molecular weight cutoff of 100 as it would 50. We also used urea in this molecular weight cutoff of 100. Urea has a molecular weight around 60. That's less than 100. It was able to diffuse. Then we jumped up to a molecular weight cutoff of 200. We did not repeat the sodium chloride portion. Why? Because we already know sodium chloride can diffuse. We did not repeat urea. Why? We already know urea would diffuse. But albumin was not able to diffuse. It is much too large. It was not able to get through those molecular weight cutoff holes. Glucose, however, has a molecular weight cutoff of 180, and it was able to move through the molecular weight cutoff membrane of 200. After all, 180 is less than 200. Moving on to activity 2. In activity 2, we were trying to learn about facilitated diffusion. And these questions that you see on the slides are the same types of questions that you saw in your review sheets in your PhysioEx, and also in the stop and think questions in your PhysioEx or the pre-lab questions. So something to consider for facilitated diffusion compared to simple diffusion. Facilitated diffusion and simple diffusion, and I'll remind you, we just addressed simple diffusion in activity 1. Their similarities include both are passive transport. That means there is no cellular energy used. No ATP is used. Rather, just the kinetic energy of the solutes. Both types of diffusion, simple and facilitated, allow the movement of the solute from a high concentration to low. And another similarity, if given all the time in the world, both will keep going until equilibrium is reached. However, on the right side of this slide, I'm noticing one huge difference between the two. Facilitated diffusion has something called transport maximum. This means there will eventually be a maximum transportation rate. What do I mean by this? As we look at our data on the next slide, we will realize that the number of carriers will be the rate limiting factor. Why is that? Another difference between simple diffusion and facilitated diffusion is that the carrier for the solute in facilitated diffusion physically binds and interacts with the solute. It changes conformation shape. It's a protein, of course, as it moves the solute from one side of the membrane to the other. So this physical interaction means it will come down to, as we increase the number of our glucose transporters, we built a membrane, of course, with these. As we increase the glucose transporters, the rate of transport increased. Let's look at that data. So again, the transport rate is in millimoles per second. Notice that we built our membranes to have 500 glucose carriers or 700 glucose carriers, or in one experiment, 100 glucose carriers. We also played with different concentrations of glucose, 2 millimolar, 8 millimolar, and 10 millimolar. In our final experiment for activity 2, we played with 2 millimolar glucose along with 2 millimolar sodium chloride. Let's look at first the 2 millimolar glucose solution. And we put built in 500 glucose carriers into the cell membrane, the dialysis tubing. So we don't need to worry about molecular weight cutoff. We're building glucose carriers. They are going to physically interact with the glucose and move them. Now, what I want you to notice that when we went from 500 glucose carriers to 700 glucose carriers, our rate of transport increased. It went from 0.0008 to 0.0010. 0.0010 is higher than 0.0008. The rate of transport increased. All right, then we tried 8 millimolar glucose. We played with this with 500 and also 700 carriers. And again, notice the more carriers we had, the greater the transportation rate. Now we played with 10 millimolar glucose and we dropped the number of glucose carriers down to 100. Notice that your data gave you a transportation rate. However, that number is useless to you. Did you notice that the system timed out before equilibrium was reached? We still had some glucose that had not been moved. Did you pay attention to that? There are even questions in your PhysioEx that ask you to address this. So we were not given all the time in the world. We were given a finite amount of time with very few carriers, a high glucose concentration, and our timer timed out before all of the glucose was moved. Now, the very final experiment for this activity, we used 2 millimolar glucose and 2 millimolar sodium chloride. We used 700 carriers for glucose. And what I want you to notice is between the first experiment and this last experiment, The rate was 0.001 for both of them, meaning the added sodium chloride did not change the rate of transport for glucose. That means sodium chloride did not compete for the glucose carriers. Those glucose carriers could only move glucose. So no competition for glucose with its carrier with another solute. Now we went to activity 3, where we were trying to simulate osmotic pressure. I'm going to remind you again that you have already been tested on your chemistry, your metric system, and scientific notation. You've already had that test. And part of that test addressed osmotic pressure, osmolality. And you had to think about dissociation factors. We also learned the difference between tonicity versus hyperosmotic or hypoosmotic. Tonicity, I'll remind you, refers to not only the ability for the solute to move or not, but also it referred to the concentration, like hypertonic. That means the outside solution was more concentrated than the inside. Okay, so and then hypotonic would refer to an extracellular fluid that has a reduced concentration than the intracellular fluid. And also the tonicity referred to that the solutes were impermeable. So we learned in our chemistry review that if a solute was found in unequal concentration across a biological membrane, and if that solute was impermeable, then water would move. Take a moment to look at the system that we were playing with on this bottom right picture. We had our 2 chambers again. We again were playing with molecular weight cutoff dialysis membranes. So now we're back to addressing, can the solute move? If the solute cannot move, what will? Water. We started out adding our solutes to the chamber on the left side, and we had water on the right side. Now, we did several experiments. We started out with a molecular weight cutoff of 20, and we played with 5 millimolar sodium chloride, and then 10 millimolar, and then we jumped up to a molecular weight cutoff of 50, etc. I'd rather go through this data showing you pictures. So here we are. Clearly, I'm not an artist, but here's our 2 chambers. And in the center, in red, I've written membrane. We had weights on top of these chambers. The weights allowed us to measure the osmotic pressure. What do I mean by that? When water moved from one chamber to the next, that chamber volume would want to expand, of course. But we applied a weight, a counter pressure, so we could actually physically measure how much pressure we had to apply to oppose the volume change. Our first experiment, like I said, we played with 5 millimolar sodium chloride with a molecular weight cutoff of 20. Please remember from activity 1, we know sodium chloride is much larger than a 20 molecular weight cutoff membrane. Water shifted from the right chamber towards that sodium chloride chamber. Particles suck. I'll remind you, if there's unequal concentration and the solutes cannot move, water will. How much counter pressure did we have to apply? We had to apply 170 millimeters of mercury. Then we repeated the experiment using 10 millimolar sodium chloride. We still used a molecular weight cutoff of 20. Water shifted again towards the sodium chloride side. But now how much pressure did we have to apply? Double. That should not shock you. We doubled the concentration. Then there was twice the amount of pull for water to move from the right chamber to the left. So of course, the amount of pressure we had to apply on our weight to oppose that volume change also doubled. Then our third experiment with sodium chloride, we played with 10 millimolar and a molecular weight cutoff of 50. There was no osmotic pressure that we detected. Why? Well, you should remember from activity 1 that the molecular weight cutoff of 50 was large enough to allow for sodium chloride to move through those pores. Why did sodium chloride move through those pores? Sodium chloride dissociates into much smaller ions. Sodium has an atomic weight of close to 23 and chloride close to 35. Both those numbers are less than 50. So if the solute can move, it will. And there was no osmotic pressure detected. Moving on through our data, we then applied 8 millimolar glucose in the left chamber, water in the right chamber, and we used a molecular weight cutoff of 100. You should remember from the first activity that glucose has a molecular weight of 180. This is much larger than 100. Glucose was not able to move. We have unequal concentrations. Water did shift. And we had an osmotic pressure of 136 millimeters of mercury. We then repeated the experiment again, but this time with a molecular weight cutoff of 200. We did not detect any osmotic pressure. Why? Because glucose could move, could diffuse through the 200 molecular weight cutoff membrane. If the solute can move, then it will diffuse, and there's no mass shifting or gross net gain or loss of water across the chambers. Now, I have a challenge question for you. In this activity, we started out playing with sodium chloride, and we played with 5 millimolar sodium chloride and 10 millimolar sodium chloride. And then all of a sudden we shifted to glucose, and we played with 8 millimolar glucose. Well, my challenge question to you is, what would have been the osmotic pressure if we could use 10 millimolar glucose and used a 100 molecular weight cutoff? We recognize glucose would not be able to move. We should know by now that there would be an osmotic pressure we detect. Now, there are two ways you can actually solve this. The first way is shown here in black. You can refer to this activity when we played with the 5 millimolar sodium chloride. When we played with 5 millimolar sodium chloride, it gave us an osmotic pressure of 170 millimeters of mercury when we used the 20 molecular weight cutoff. Okay, well, sodium chloride has a dissociation factor of 2. So again, when we played with sodium chloride 5 millimolar using the 20 molecular weight cutoff, sodium chloride couldn't move, and we detected or had an osmotic pressure of 170 millimeters of mercury. So my challenge question is, if I use 10 millimolar glucose using 100 molecular weight cutoff, glucose cannot move, cannot diffuse. Glucose has a dissociation factor of 1. So 10 millimolar glucose would give me a 10 milliosmolar. So that would also give me an osmotic pressure of 170 millimeters of mercury. But there is a second way you could have solved this problem using proportions. And I'll remind you again, you have already taken your math and chemistry quiz. All you had to do was use proportions. If 8 millimolar of glucose gave us a pressure of 136 millimeters of mercury, then what would 10 millimolar glucose give us in millimeters of mercury? All you have to do is cross multiply and solve for X, and you would still get 170 millimeters of mercury. Our last experiment, we played with a molecular weight cutoff of 200. We had 9 millimolar albumin on one side. My albumin is shown here with the magenta or pink dots. On the right side chamber, we had 10 millimolar glucose. I'll remind you that glucose can diffuse through a molecular weight cutoff membrane of 200. Glucose is represented in this picture of mine as green dots. Because glucose can move, it diffused from a high concentration to low. It diffused until there were equal amounts of glucose in the right side chamber as there are in the left side chamber along with albumin. So now the left side chamber has not only the 9 millimolar albumin, but also about 5 millimolar glucose added to it once the glucose diffused. So which side would have the highest osmotic pressure? The side with the albumin, and also now including the glucose because the glucose diffused. I bet you need some more help with that concept. I'm glad you do, because I am now going to tell you what you can expect on your lab exam for this particular activity. For lab exam number 1, you are responsible for remembering all of the molecular weight cutoff membranes we played with in activity 1 and in activity 3. You are also responsible to know the concept of osmotic pressure, which means you need to remember your dissociation factors. Well, sodium chloride has a dissociation factor of 2. Urea is amino acid based, so it does not dissociate. Albumin is a very large protein. It does not dissociate. And glucose, a sugar, does not dissociate. Why? Urea, albumin, and glucose are all their individual atoms in those molecules are covalently bonded. So let's practice with some examples. So there's an example here on the screen, but I'm going to give you some more specific examples on the next few slides. Your possible test questions, you will have word problems where I will describe on the left chamber, you have this solution. On the right, in the right chamber, you have this solution. Which chamber will have the highest osmotic pressure? Just like we did in this activity. Okay, you must ask yourself this main question in order to answer all these problems. Can any of these diffuse? Can they get through the molecular weight cutoff? If both solutes can diffuse, then there is no osmotic pressure that will be detected. If the solutes can move, they will. There will be no net gain or loss of water. But what if only one can move? Well, if only one can move, then the osmotic pressure will be higher on the side of the solute that can't move, just like we saw at the end of activity 3 with albumin and the glucose example. Glucose could move, but albumin could not. But what if neither of the solutes can move? Then you must consider dissociation factors. So let's use some examples. Here we have a solution of 5 millimolar sodium chloride in the left chamber and 5 millimolar glucose in the right chamber. We have a molecular weight cutoff of 50 dividing the two chambers. And I'm using my little hand here to simulate a weight. So we ask ourselves, can any of these solutes diffuse? Well, we recognize sodium chloride can diffuse. It can fit through the molecular weight cutoff of 50, but glucose can't. So in this picture, sodium chloride is represented as green dots and glucose as the magenta dots. And you can see that sodium chloride will move until there are equal millimoles on in both of the chambers. Because the right chamber will now have 5 millimolar glucose plus some moles of sodium chloride, the water will shift from the sodium chloride side to the glucose now mixed also with sodium chloride. So the osmotic pressure is higher on this glucose sodium chloride mixture chamber. Notice the weight is now pressing down to counteroppose that volume change. Here are two more examples. In the top picture, we have a molecular weight cutoff dividing two chambers of 20. In the left chamber, I have 5 millimolar sodium chloride. In the right chamber, I have 5 millimolar glucose. Ask yourself the question, can either of these solutions diffuse through the membrane? The answer is no, neither can move, neither can dissociate. Well, now we have to move to the next consideration, which is dissociation factor. You can see on the left side that sodium chloride has a dissociation factor of 2, which means 5 millimolar would be 10 milliosmolar. In the right chamber, you see that I've written that glucose has a dissociation factor of 1, which means 5 millimolar glucose will still be 5 milliosmolar glucose. Now, which side has the higher particle count? Clearly, the sodium chloride side does. So particles suck. If neither of these solutions can move, and we clearly have unequal osmolarity, the water will shift to the side with a higher osmolarity. So that means water shifts to the left chamber. The sodium chloride chamber will have the higher osmotic pressure. Now look at the bottom picture. We have two chambers separated by a molecular weight cutoff membrane of 200. In the left chamber, I have 5 millimolar sodium chloride. In the right chamber, I have 5 millimolar glucose. Now, I ask the question, can either of these move? Can either of them diffuse? Both can. Both can easily get through those molecular weight cutoff pores. So if both can move, if both can diffuse, there will not be a net gain nor loss of water in either chamber. That means no osmotic pressure. Here's another example. I have a molecular weight cutoff of 100, separating a left chamber that has 5 millimolar glucose and a right chamber that has 5 millimolar albumin. I ask the question, can either of these solutes diffuse? The answer is no. So when the answer is neither can move, I must move to the next consideration, which is dissociation factor. Both of these solutes have a dissociation factor of 1, which means both of them will have the same 5 milliosmolar solutions. There is no difference in the solute concentrations. True, they are very different solutes, but they have the same number of particle count in both of the chambers. So there will not be a net gain nor loss of water between either chambers. No osmotic pressure will be detected. Let's do one more before we move on to activity 5. Here's a challenge one. Let's say you have a molecular weight cutoff of 20. I have a 5 millimolar sodium chloride in the left chamber, and I have a 10 millimolar glucose solution. Now, I ask the question, can either of these diffuse? And the answer is no. And now I must consider dissociation factor. Well, sodium chloride has a dissociation factor of 2. So 5 millimolar times 2 will give me 10 milliosmolar. Glucose is at 10 millimolar, and it has a dissociation factor of 1. So that means 10 millimolar of glucose will be 10 milliosmolar. Notice both sides of this chamber, both chambers have the same concentration, the same milliosmolar concentration. There is not going to be a net gain nor loss of water. There will not be any osmotic pressure detected. So that is a challenge one to represent, to show you that you must consider your starting millimolar concentration and also consider dissociation factor in this example. Let's move on to activity 5. In activity 5, the data you collected is actually meaningless. What you should have paid attention to, if you look at the bottom picture here, was your apparatus. What you should have paid attention to was the amount of ATP, your starting concentration in the cell versus the beaker, and your finishing concentrations. And also, one more thing that PhysioEx did not tell you to write down was the rate of transport. Thankfully, I took screenshots of all of the 5 activities, 5 experiments, I should say, that you did in activity 5. So in this activity, the important part was to understand how the sodium potassium ATPase works. Sodium potassium ATPase, also known as the sodium potassium pump. Pumps require cellular energy. That's the ATP that you're seeing here in this video. And pumps will move at least 1, or in this case, 2 different solutes against their concentration gradient, meaning they're going from low to high. So notice potassium is being moved into the cell where already there is high potassium inside the cell, shown here as the green circles. And sodium is moved from inside the cell to outside the cell, where sodium is already in high concentration. Also notice that it is unequal movement of these solutes. 3 sodium move out for every 2 potassium that move in. Again, we were going to build a membrane very similar to what we built in activity 2 when we were building glucose carriers in that activity. We did 5 different experiments in this activity. We built sodium potassium pumps into our membrane. Notice that we have electrodes that stick into the cell and also electrodes that are outside the cell. And eventually, we also build into our cell membrane glucose carriers. The data you collected probably looks like this. And you can again see that we did 5 independent runs. 1, 2, 3, 4, 5. And you collected your data, and your data came out in a chart like this, where the very last column gave us the rate of movement in millimolar per second. This data is totally useless. This table is completely useless. It does not show you what you should have seen. What you should have paid attention to were the pictures in each of these 5 independent runs. So again, I took screenshots and I'm going to go through them. I urge you to listen to this part of the video many times until you understand this concept. Again, we're playing with the sodium potassium ATPase. It is a pump. It requires cellular energy. It moves 3 sodium out of the cell for every 2 potassium that are moved into the cell. And I also want you to pay attention that there is a physical interaction between this purple protein that you are seeing here that's actually moving these solutes, a physical interaction. So this is going to mean that we're going to have a Tmax, a transport maximum, and the ultimate take-home message for this activity is to recognize that the rate limiting factor is the number of pumps we built into the membrane, not the amount of ATP. Nope. The number of pumps we built into the membrane. Again, the data you actually collected and wrote down, totally useless for testing purposes. So pay attention. In run number 1, I want you to notice that we had 9 millimolar sodium chloride on one side and 6 millimolar potassium chloride on the other side. 9 millimolar to 6 millimolar. That's a 3:2 ratio. Why is that 3:2 ratio important? Look at how this pump works. It moves 3 sodium for every 2 potassium. So that 3:2 ratio for our starting cells and beakers, very important. We also need to pay attention to how much ATP we added. In this first run, we had 1 millimolar ATP. Now that's the fuel, that's the energy that the pump needs to move the sodium from one side to the other and the potassium from the other side. Now here's the part you did not write down, and here's the part you probably not pay attention to. Notice up here where I have my red magenta star. We went from 9 millimolar sodium chloride on one side down to 5. And for potassium, we went from 6 down to 3.742. This did not run to completion. We still had sodium chloride on the cell side that we could have moved, and we still had potassium on the other side that we could have moved. The rate of transport was 3 minutes. What does that mean? It took 3 minutes and then the system ran out of ATP. We ran out of ATP. So we could not finish moving all the 9 millimolar sodium chloride from the cell side to the beaker side, and we could not move all the potassium chloride from the beaker side to the cell side. So that's important. Okay, so now we say, let's repeat it. And this time we used 3 millimolar ATP. We had the same number of pumps built in as the first one. We had 500 for the first run. We have 500 for the second run. And the data you wrote down over here, the rate in millimolar per second, if that's what you pay attention to compared to rate 1, you actually get a slower rate. And yet the most important part is what you missed. The rate of transport was 60 minutes. We went for full 60 minutes, and notice that we completely moved all the 9 millimolar of sodium to the other side, and we moved all the 6 millimolar potassium to the opposite side. We went to completion. That's different than run one. Run one, we ran out of ATP after 3 minutes, and we did not get to full movement of these solutes. Okay, so now we recognize having enough energy is important, but is that the most important part of active transport? Okay, well, let's repeat this again with 500 sodium potassium pumps. We have our 9 millimolar sodium chloride on one side. But notice we don't have our 9:6 ratio. We didn't put potassium chloride on the other side. We had enough ATP. We did not get any rate of transport, none. Why? This protein pump will not function unless it has this 3:2 ratio. If it could talk to you, it would say, oh, heck no. I'm not wasting energy just to move sodium ions. I want this bus fully loaded. I need to move 3 sodium for every 2 potassium. If I do not have that, I'm not working. So that's why we did not have a rate of transport. Okay, so now we recognize we need enough energy. We need this 3:2 ratio, 3 sodium for every 2 potassium. Okay, all of those things sound very important, don't they? Well, now let's do run 4. Notice that we increased to 800 pumps. Look at your picture up here. Again, this is the data you should have written down. Notice we used 3 millimolar ATP, and notice we moved all of the sodium from the cell to the beaker side and all of the potassium from the beaker to the cell side. Moved it all. We moved all of the potassium from outside the cell to inside, and all the sodium from outside the cell, or from inside the cell, sorry, to the outside, to the beaker side. And again, if you look at the rate of transport, it's very misleading. The rate in millimoles per second. What was most important was the little sign that showed up on your PhysioEx that said the full rate of transport took 36 minutes. Okay, so now let's talk about that. We had 3 millimolar ATP. We had our 3:2 ratio, and now we had 800 pumps. Compare that to run number 2, when we only had 500 pumps. That experiment went to completion, but it took 60 minutes. This time with 800 pumps, it only took us 36 minutes. So everything else stayed the same. 3 millimolar ATP, enough energy, check and check. A 3:2 ratio of 3 sodium for every 2 potassium, check and check. And yet our total time for the rate of transport was almost half. 36 minutes it only took us to reach completion. And that, my friends, is the number one most important thing. Oh, that's a very sad star. Let's do that again. It's the number of carriers that is the rate limiting factor. In this case, the number of pumps. You increase the amount of sodium potassium pumps, those pumps will be able to move your 3:2 sodium to potassium ratio much more quickly, assuming all the energy is there required. And that I am 100% sure you missed when you did this activity for PhysioEx number 1. I'm sure you missed it. And yet those little signs came up and told you rate of transport, total time, and PhysioEx never told you to write it down. So it's really not your fault, my young Padawan learners. You weren't directed to write that important piece of information down. The part you were told to write down, meaningless. That data means nothing. You need to know what you saw, what you did, how you manipulated each of these runs, and it's the number of pumps that was the rate limiting factor. Our last run, we again played with 3 millimolar ATP. We again had a lot of pumps for our sodium potassium ATPase. We had 800. We know that's really good. But this time we built into our membrane also 400 glucose carriers, 400. And we were using 10 millimolar glucose. And you'll notice that when the system timed out, it timed out on us at 36 minutes. That's the same thing that we saw for the sodium potassium movement in run number 4. So this was run number 4. But notice that the glucose, see this little pound sign right here? The glucose was not able to move. It was not, those glucose carriers did not reach equilibrium for the glucose because there weren't enough carriers to move the amount of 10 millimolar glucose until it reached equilibrium. The system timed out on us. That's something that we saw also in activity 2, if you recall, when we played with 10 millimolar glucose and we didn't have 700 carriers, remember? So we were very short on glucose carriers. Now, the important part on this was to show that glucose in the mix did not impair the sodium potassium ATPase. Notice the rate of transport for the sodium and potassium was still 36 minutes, the same that we saw in run number 4. What does this mean? Glucose does not compete for the pumps, and the sodium and potassium did not compete for the glucose carriers. Okay, take-home message for both facilitated diffusion and active transport. There is physical interaction of the solutes with those protein carriers or pumps. And it's the number of carriers / pumps that are the rate limiting factor. Another thing to remember from our earlier activities, between activity 1 and 2, is simple diffusion and facilitated diffusion have a lot of similarities, but there are also important differences. And the next thing to remember for your lab exam, in addition to your data and the importance of the data that I went through, is you need to remember your molecular weight cutoff membranes that we played with and the solutes that we played with. When could those solutes diffuse and when could they not? And I'm, of course, referring to activities 1 and 3 for those types of concepts. And again, you can review these possible test questions that I gave you some examples for. Okay, thank you for joining me for PhysioEx number 1.